Now we are going to start with the antibodies. So first of all, what is the definition of antibody? Antibody can be defined as the specialized glycoproteins which are produced from the plasma B cells against an antigen. This is the whole definition of the antibody. So antibodies are specialized glycoproteins which are produced by the plasma B cells against a particular antigen. Those are called as the antibodies. Now there are five types of antibodies which are IgG, IgA, IgM, IgD and IgE. Now let's read about the ultra structure of the antibody. Although this is not generally asked in any of the exams at UG levels, but still you should have the basic idea of the structure of the antibodies. So see here, the antibodies looks somewhat like Y. Okay, it somewhat looks like a Y. So it is a Y-shaped heterodimer. Why it is a heterodimer? The heterodimer uh, word meaning that means it is made up of two different types of chains. One is heavy chain and the other one is the light chains. Okay, so there are total four polypeptide chains, out of which two are heavy and two are light. See here, this is a light chain, then this is a light chain, and this one is here is the heavy chain, and again here this one is the heavy chain. So it is made up of two light chain and two heavy chains. Two light and two heavy chains are there in any antibody. Now these H and L chains, the H for heavy chains and the L for light chains. These chains are bound together by disulfide bonds. See here, everywhere you will be able to see that disulfide bonds. So see here that here there are disulfide bond. Then between the heavy and the light chains there is disulfide bond. Here also between heavy and light chain there is disulfide bond. So they are all connected through the disulfide bonds. So there are five classes of heavy chains. Those based on those five classes, there are five types of antibodies. So the five classes of heavy chains are the gamma heavy chain, alpha heavy chain, mu heavy chain, delta heavy chain, and the epsilon heavy chain. Now, when the heavy chain present in the antibody is a gamma heavy chain, then it will be called as the IgG antibody. If it is a alpha heavy chain, then it will be called as IgA antibody. If it is mu heavy chain, then it will be called as IgM antibody. If it is delta, then it will be called as the IgD antibody, and if it is epsilon uh, class of heavy chain, then it will be called as IgE antibody. So depending upon the heavy chain, the class of heavy chain, they the antibodies are classified. Based on that class, the naming of the antibodies are given or naming is done. So this is based on the different amino acid sequences in the heavy chain. Okay, this classification between gamma, alpha, delta, mu, epsilon heavy chain is based upon the different amino acid sequences in the heavy chains. Okay, now the light chains are also of two types. One is the kappa light chain and the delta light chain. So in any antibody at a time, only one type of light chain will be present. That means if one light chain in suppose IgG antibody is kappa light chain, then the other light chain would also be kappa only. It cannot happen that both kappa and lambda light chains are present simultaneously in a antibody. Okay, now come to the important point of variable region. So what is variable region? Variable region is that region of the antibody which varies time to time based on the antigen. Okay, so this area is the variable region. This is nothing but the initial 110 amino acid of the at the ns3 terminals of the heavy and the light chains. So the first 110 amino acids at the ns3 terminal of the heavy and the light chains makes up the you know the variable region. Okay, so see here, this is our the amino terminals. This is our amino terminal. This is our amino terminal of heavy chain. This is amino terminal of heavy chain, and this is amino terminal of the light chain. Okay, so here this region is making this region consists of 110 amino acids, and here also this region consists of 110 amino acids in both the light chains and the heavy chains, and that makes up the variable region of the antibodies. Okay, and that variable region is for the binding of the antigens, hence that portion is also called as the antibody binding fragment. So that is called as antibody binding F for fragment. So antibody binding fragment or fab. Okay, now come to the hinge region. What is hinge region? Hinge region is the junction region. This region, this area is called as hinge region. Okay, let me clear it so that you can understand it better. So this area, this area is called as hinge region. This area is called as hinge region. Okay, see here I have written also, this area is called as hinge region. This hinge region is very flexible area. Why is that flexibility is there in that antibody? That is because of the proline and the cysteine amino acids being present at that point. You must know from your biochemistry knowledge that these amino acids provide flexibility in the amino acid chain or the amino acid polymer. So since those antibodies have this proline and this cysteine amino acid rich in that area, hence that area is very much flexible and is called as the hinge region. So this is in short all about the antibodies. Okay, this is in short all about the antibodies. If anytime it comes in your exam to write a short note over any antibody, then you must make the diagram and then describe everything in short. Okay, everything, these all things should be described in short. Okay, now come to the enzymatic digestion of the antibody. Now this is not generally asked, but may be a question in your MCQs in your university exams. So you can just go through it once before going to the exams for 1 marks of the MCQ if it is asked in the MCQs in your university exams. Okay, so what is the enzymatic digestion of the antibody? So three types of digestion can occur of the antibody, that is by the papain digestion, the pepsin digestion, and the mercaptoethanol reduction. So what happens when there is we apply the papain in the antibody, then what happens is that this fragment and this fragment gets separated, while this fragment gets separated. So there are total three fragments with the papain. With the papain there are total three fragments formed, two FAB fragments and one FC part. Okay, so this is the two FAB fragments, this is one fragment, and this is another FAB fragment, and this is the FC part. So there are total three fragments, and the names of the fragments are two FAB and one FC fragment. Okay, and when we are having this pepsin digestion, when we have the digestion by the pepsin, so pepsin breaks the bond here. Okay, pepsin breaks the bond. Let me clear it so that you can understand. So pepsin makes the break here. Okay, pepsin makes the break here, so that these two fragments remain together. These two fragments remain together, and hence it is called as AB twice. FAB twice. What does that mean? That means that antibody fragment part, I mean antibody binding part. Okay, antibody binding part is two times. I mean two number of antibody bindings are present in that. One is this antibody binding part and the other one is the this one of the antibody binding part. So there are two antibody binding portion or the fragments in the same fragment of the antibody. Okay, hence it is called as FAB twice, antibody binding twice. Okay, FAB twice. While this fragment which gets separated, this fragment gets separated, this is broken in multiple smaller fragments which are called as the FC fragments. Okay, now come to the mercaptoethanol reduction. So mercaptoethanol, what does that do is that the mercaptoethanol breaks down all the you know uh, disulfide bonds. So mercaptoethanol breaks down all the disulfide bonds. As a result, what happens that all these heavy and light gels get separated from each other. So this bond is broken down, this is broken, this is broken, and this is broken. So there all together there becomes four polypeptide chains, that is two heavy chains and the two light chains are formed. So this is one heavy chain, second heavy chain, one light chain, and the other one light chain. So since due to in breakage of those disulfide bonds, there are total four polypeptide chains formed after mercaptoethanol reduction. So please remember this enzymatic digestions, that is papain digestion, the pepsin digestion, and the mercaptoethanol reduction. This is all about the first part of the antibody chapter. There is not much to remember from here, but you can remember this part because this may be asked in the MCQs, and this part is very easy. This structure of antibody is very easy if you remember this structure of antibody, then you will be able to write all these things. Okay, this is very easy chapter. Okay, that's all for the first part. Next we will talk about different types of antibody in the second part.